Gene-environment interaction describes how genotypes, our unique genetic code, develop differently according to the environment, producing different phenotypes, the physical expression of our genes. To visualize gene-environment interaction, we can use a graph with genotype along the x-axis and phenotype on the y-axis. We look at the relationship between genotypes and the resulting phenotypes, depending on the environment. To illustrate this, we'll look at 3 different example environments. In environment 1, we can see a positive relationship between genotype and phenotype. We might say that environment 1 is favourable for this phenotype because it facilitates the expression of the genotype. In environment 2, the phenotype does not change as the genotype increases, so this environment does not have an impact on the expression of the genotype. And in environment 3, we can see that the phenotype decreases as the genotype increases, suggesting that this environment is adverse for the expression of the genotype. Let's use the example of musical ability. If someone has a high genetic propensity for musical ability, we might say that they have a natural talent for music. And we know that when someone practices music a lot, their musical ability is likely to increase. So the environment has an important influence too. But as we saw before, the expression of the genotype depends on the environment. So if a person with high genetic propensity for musical ability grows up in a musical environment where they play and listen to music often, they would likely develop excellent musical ability. However, if that same person with the same genotype for musical ability grew up in a different environment and focused on different skills, for example, sports, the development of musical ability would be less supported than it was in the musical environment, and so the resulting phenotype would be weaker. Let's go back to our graph, with genotype on the x-axis and our phenotype on the y-axis. The musical environment is favourable for the expression of the genotype, so there's a strong positive relationship between the genotype and the phenotype of musical ability. The non-musical environment does not facilitate musical ability as much as the musical environment does. We can see the gene-environment interaction in the difference between the 2 environments at low and high genotype levels. There's a much bigger difference when the genotype is high, meaning that the genotype is expressed differently according to the environment. In a gene-environment interaction, the interplay of 2 variables, in our example musical ability and genotype, results in more than their sum, meaning that the effects are not solely additive. Additive effects would look like the graph on the right here, where the difference between the 2 environmental conditions remains the same as the genotype increases. Here at the Hungry Mind Lab at the University of York, we're researching gene-environment interaction in early life cognitive development. We want to find out how important gene-environment interactions are for children's early development. The development of children's cognitive skills in the first few years of life affects school readiness, educational attainment, and a variety of other outcomes later in life. There are some key challenges with studying gene-environment interactions. Data from large populations are needed to observe these interactions, because the effect sizes are very small and difficult to detect. Also, there are a range of different environments. We are including as many environments as we can in our analyses to identify the ones that interact with genotypes in early life cognitive development. So, why are we doing this research? Understanding the role of gene-environment interaction in early life cognitive development could help us to identify those who would benefit from extra support and provide information to deliver targeted and personalized interventions.